welcome to everyone. My name is Jen Whitside. I'm a director in Alumni Relations. Thank you so much for joining us today for this webinar and for all the virtual programming that you have been registering for and logging into lately. We love seeing everybody from across the country watching the programming. Today we are going to start with an introduction from Marian McCord. She is our moderator for today's webinar. She joined UNH as the Senior Vice Provost for Research, Economic Engagement and Outreach in February 2020. McCord came to UNH from North Carolina State University, where her 25-year career spanned faculty, research, and administrative roles. Most recently, she was the Associate Dean for Research at NC State's College of Natural Resources, where she oversaw the college's sponsored program portfolio and research infrastructure with more than 200 active projects and 19 testing and service centers. McCord received an SCB in Biomedical Engineering from Brown University, an MS in Bioengineering, and PhD in Textiles and Polymer Science from Clemson University. So with that, I'm going to turn it over to Marian. Thank you.
Thank you, and welcome to all our alumni and community listeners. We're really happy to have you here. We know these are incredibly challenging times, and we're glad you're with us today. So I'm Marian McCord, and I arrived in February just in time to shut research down at the university, but since then we have been able to get it started back up, and we're looking toward the future and starting to allow more and more of our research to progress in our labs. I want to introduce you today to our two presenters. First, we have Tim Sheahan. Tim's a 1999 graduate from UNH's College of Life Sciences and Agriculture. He's currently an assistant professor in the Department of Epidemiology at UNC Chapel Hill. Dr. Sheahan is an expert virologist whose research is focused on understanding emerging viral diseases and developing new means to stop them. He was previously at Harvard Medical School and Mass General Hospital before moving to pursue graduate studies at Chapel Hill about the original SARS coronavirus. After completing his postdoctoral fellowship at the Rockefeller University and work at GlaxoSmithKline, Dr. Sheahan returned to UNC, joining the faculty in 2015. Dr. Sheahan is currently engaged in multiple interdisciplinary research projects to address the immediate public health emergency of COVID-19. Speaking after Tim will be Kelley Thomas, who's the director of the Hubbard Center for Genome Studies, a leader in comparative and environmental genomics here at UNH. Kelley is also a professor of molecular, cellular, and biomedical sciences here, and he's the scientific leader of a university team that worked around the clock to successfully launch a cutting-edge COVID-19 testing lab on campus in Durham. This lab has been up and running since the students returned to campus and recently received national attention when the Coronavirus Response Coordinator, Ambassador Deborah Birx, visited campus. You should know that you should ask your questions in the Q&A box at the bottom of the screen. Do not use the chat box because that will only be available internally to the speakers. So please direct your questions within the Q&A box. And with that, I'll turn it over to Tim.
Thank you, Dr. McCord. This is very exciting. Hello, Wildcats. I'm happy to be to have this invitation from Jen Woodside to talk to you today about some of the things that we've been doing at UNC Chapel Hill since we figured out this virus that emerged in China in December is a coronavirus. So I want to tell you a little story about how we've been preparing for tomorrow's pandemics today through the development of broad spectrum antiviral drugs. But I first want to start it off with a little bit of information about myself and general coronavirus biology before I get into some of the things that we've been doing to address the pandemic. So I came to UNH from Rhode Island. I grew up in Rhode Island. So for some reason, I still had my student ID in a box at my house. It was issued back in the day when your social security number was your student ID, which I imagine you don't do anymore. So I had to fuzz that out. But I've been playing guitar forever, and you know, I was in this band in college that was featured in the New Hampshire. This is a clip from the New Hampshire on the right from 1997, playing at the Elvis Room, which is now a defunct club in Portsmouth. But it was an amazing place, and so I was really focused on music and natural resources biology. So I came to UNH as a water resources management major, or WARM major, which I don't think exists anymore. And I really learned like how to do science and set up experiments from Bill McDowell, who was leading the program at the time. But because, you know, water pollution is a major component of the education of a WARM major, I had to take microbiology as a prereq. And that was taught by Louis Tiza at the time. And that really opened my eyes to this microscopic world that's all around us. And that microbes had the power to make people really sick and even kill them. And I gained practical lab experience at the Jackson Estrine Laboratory with Steve Jones. And one of my favorite classes was a cell culture class taught by Tom Foxall, which I took later on in my career, which really positioned me to get my first job in academia as a technician after leaving UNH. So basically, being able to grow mammalian cells in culture is the foundation for lots of molecular biology and cell biology. So like learning that skill essentially got me my first job. So thanks, Tom, for that. So today I want to talk to you about emerging viruses. And so lots of viruses circulate among wild and domestic animals. And sometimes they enter the human population through what's called a spillover event. And if that virus is able to replicate in a human and be transmitted from person to person, this could cause a local outbreak or epidemic. But it's so easy to get on a plane and travel these days, so it just takes one sick person getting on a plane to take this virus across the world, leading to a global epidemic or even a global pandemic. And so I made this cartoon a couple of years ago, and what we just saw basically has played out in real time over the past 9 months with the emergence of SARS-CoV-2, the causative agent of COVID-19. So emerging viruses and newly emerging viruses, they're new in a sense that they're not known to have previously infected or caused disease in humans. So they're not new to Earth, they're just new to us. So they exist in some other reservoir species or some other animal, and they jump into humans to create something new, like COVID-19. So I'm part of a pretty big group. It's a group of about 30 people now. It's headed up by my PhD advisor, Dr. Ralph Barrick, the guy in the mustache in the middle. So this is postdocs and grad students and technicians and staff. And we have been working around the clock like a finely tuned machine since January 11th, when we found out that this new virus that had emerged was in fact a coronavirus. So what is a coronavirus? And given Marian's experience with Latin, she can probably answer this question. Coronavirus get their name from the Latin word for crown, which is corona, because of the spike-like projections that stick out of the virus particle. So this is an electron micrograph of the original SARS coronavirus taken in about 2002, 2003. And you can see these spike-like projections coming out, which give the coronavirus family their name. And coronavirus as a family, they have what we call emergence potential. So this cartoon is showing all 7 of the known human coronaviruses on the right. And they were originally, all of them were originally thought to have emerged as zoonoses. So basically they were viruses that infected some other animal and jumped into humans to create a human disease. For example, OC43 is very closely related to a virus that infects cows. And this jumped into humans in the 1800s, it's thought, to create a common cold-causing coronavirus called OC43. And I don't really need to stress this point, but you know, there's definitely a need for coronavirus therapeutics. In fact, over the past 20 years, 5 new coronaviruses have emerged, 3 of which cause new human disease. The first one in 2002 with the original SARS coronavirus, then in 2012 with MERS coronavirus, and lastly with SARS-CoV-2, which causes COVID-19 in 2019. But this is not a uniquely human situation, and 2 of the 5 new coronaviruses to emerge in the past 20 years make pigs really sick and cause the deaths of millions of piglets every year. So coronavirus as a family in general, they like to jump from one species to another, and we need to start preparing for this eventuality because I think this is going to continue to happen in the future. So one way that we've been trying to do this is through the development of broad spectrum antivirals or therapeutics. And typically, if you're going to develop a drug, the typical strategy was we're going to create one drug for one bug. So we have MERS coronavirus. We want to make one drug that works great against MERS coronavirus. But in making this very specific drug, you may basically engineer away breadth. So it might not work against other coronavirus, but it might work great against MERS. So what we've been doing to get around that is develop broad spectrum therapeutics. So we have one drug for many bugs. So we want this drug to work against not only the coronaviruses that we know about today, but those that may emerge in the future. And over the past 6 years now, we've been working with this interdisciplinary and interinstitutional team below that includes big pharma and biotech and other academic partners. And so at UNC, we have special laboratories full of special people to work with these deadly viruses. So I'm on the left-hand side at what's called biosafety level 2. I'm wearing a lab coat and gloves and safety glasses, which I wear all the time. I'm handling my virus in what's called a biological safety cabinet. This is a glass-fronted cabinet that helps contain the virus within this physical space and prevent any contaminants that might be floating around my room from getting into my virus cultures. On the right-hand side is my colleague, Dr. Amy Sims. She's at biosafety level 3. So this is a high security, high containment lab. And in this lab, you wear a full Tyvek suit with an apron, double gloves. You have a hood that's connected to a battery-powered air purifying respirator feeding sterile air into her hood. So she's not breathing the room air. She's breathing sterile air provided by her respirator. And like me, she's doing all the virology within the safety cabinet to help keep the SARS and the MERS and the SARS-CoV-2 contained in that space. And so since 2012, we've been doing a lot of preclinical drug research. So I'm just trying to show you the arc of a vaccine or a therapeutic, which has a preclinical stage and a clinical stage. It usually takes 10 to 20 years and costs a ton of money. And so what we've been doing is kind of working on this end, trying to figure out if a given drug will have any efficacy against a given virus. So we work in coronavirus. So we look in small animal models and in cell culture models to see, like, will the drug work or not? And after these hoops are jumped through, you go through the various phases of human clinical trials where you're looking at the safety and tolerability of a drug as well as whether or not it's going to work in people. And so we have this, I think, kind of unique and comprehensive platform to preclinically evaluate antivirals. So we actually have the ability to create viruses in the lab such that if someone isolates a virus from a bat in Maryland or Concord, New Hampshire, and they publish that sequence, we can then create the virus in the lab from the sequence alone using the genetic tools we have in the lab, and then test various drugs or vaccines in models that we build with this new virus to see whether or not these drugs or vaccines will have an effect on diminishing virus replication. And one of the cool models that we have is called a primary airway epithelial cell culture model. So this is a model of the human conducting airway, or the cells that line your windpipe. And if you look at a cross-section of the culture, it looks just like the cells in your windpipe. So you have ciliated epithelial cells on the top, you have mucus secreting cells, so you actually have to wash the snot off the surface of these cultures every few days because these cultures can't clear their throat or cough. And recently, Kate Edwards in the lab infected these cultures with SARS-CoV-2, fixed them with a chemical to make them safe to remove from the laboratory, and then gave them to a woman named Camille Eyre, who's a talented electron microscopist. And she took a picture of the surface of these cells that were infected with SARS-CoV-2. And each one of these little balls is basically a virus particle. And so you see tons and tons of this virus particles coming out of the surface of these cultures. So basically, this is what your airway looks like when you're infected with this virus. And so that's why when you speak or you cough or you sneeze, you're basically projecting this virus out, making it available to be inhaled and potentially infect another person. So one of the drugs that we've been focusing on is called remdesivir. You might have heard about it in the news. It was originally developed as an Ebola virus drug, shown to work in cell culture models and in monkey models for Ebola virus hemorrhagic fever. And it was actually tested in human clinical trial in the latest Ebola outbreaks in West Africa and in the Democratic Republic of Congo. It was actually given to the first ever neonatal survivor of Ebola virus hemorrhagic fever. So this baby was born in Guinea to a mom who had Ebola was born, took some medication, and ended up clearing the virus and surviving. So that's kind of an amazing story. But we wanted to know, will this work against coronavirus? So in 2015, we started this work. We started testing it against SARS-CoV-1, MERS, lots of different coronavirus from pigs and bats, as well as common cold causing coronavirus. And when SARS-CoV-2 emerged, we tested it against that. And we figured out that it basically works against all coronavirus that we know about, that we have in the lab. And because of this preclinical work, it basically positioned this drug to be immediately deployed in clinical trial in humans in China and in the United States and elsewhere, and ultimately led to the approval of this drug last week by the FDA. Another promising antiviral that we're working on is called EIDD2801, and unlike remdesivir, it's an oral drug. Remdesivir is an IV drug, so if this ends up being safe and efficacious in people, this could really be a game changer, because you could just go to CVS and you could get this medication that your doctor prescribes, and you could take this at home rather than remdesivir, which is really only able to be used by the most sick patients who are hospitalized. And this is now in phase 2 clinical trial at UNC Hospital, so that's really exciting. And one kind of strange thing that I hadn't anticipated being a coronavirologist during a coronavirus pandemic is that I went from being asked to comment in various news sources about what coronaviruses are and their biology to being the news, where GQ magazine wrote a profile about a virologist. So basically, like, for some reason, the life of a virologist is something that might be of common interest enough to make a story about it. So that was definitely shocking, and a lot of fun to see scientists, you know, on the forefront of the news, which they hadn't really been like they are today. So I'm going to just wrap it up here and just reiterate that I think coronaviruses as a family, they have emergence potential, and we need to be doing things today to not only address the COVID-19 pandemic, but also to prepare us for the potential emergence of COVID-20, 25, 28, 29, 30, whatever. So thank you for listening. Thank you to the National Institutes of Health and the North Carolina State Legislature for our funding, and all the wonderful people that are listed on this slide that have contributed to the work that I talked about today. At this time, I'd like to invite Dr. Kelley Thomas to talk about the wonderful testing efforts that are underway at the Durham campus of the University of New Hampshire, and what you've done to not only bring your students back, but to keep them safe.
Thank you very much, Tim. That was a great talk. As Tim mentioned, I'd like to just use a few slides to talk about this laboratory that we've set up here on campus and how that process went to do that. I'm using a set of recycled slides. I had an opportunity a week or so ago to hold a workshop with other regional core facility directors and people that manage like genomics core like ours here at UNH, and talk specifically about testing at universities, and got a chance to meet with some of the other universities that are trying to stand up their own laboratories and how they're doing it. So I was just going to take this time to talk about how we set this up a little bit. We have Marian McCord here, of course, who is one of the leaders. I'm so glad that she she came when she did. She may not be as glad, but it's been a very important thing for us to have that leadership. So if we can have the next slide. Yeah, so just very briefly to talk about the testing strategy, and without getting too far into the weeds here, but really, when the university had to close last semester, there was a concerted effort to try to understand what we could do moving forward. And so one of those things was to establish a set of task forces, and the one that I became involved in, I wasn't involved in right from the beginning, was a testing and tracing task force that Marian and Chief Dean were leading, and to talk about you know, what can we do going forward. There was a decision that was made early on that if possible we should try to do testing on campus, and that this team of people was really made up of both administration people and faculty people from all sort of walks of life here on campus, and with all different kinds of expertise, which was really fundamental to being able to pull this off. And a lot of hard work, I have to say. My involvement has really been just on the design and establishment of the laboratory itself and its capabilities, which evolved over time, certainly. So just to talk a little bit about how the lab operates and how it accomplishes what it does, there are really two sort of fundamental things if you want to test a large number of samples. And one is depicted by this picture on the bottom here. What you're seeing on the left, this is how the samples arrive at the lab. It's in giant garbage bags full of samples, and somehow, and this seems like a trivial thing, but somehow, thousands at a time, they need to be moved from that pile of samples. They need to be taken out of the bags and processed, and then they end up in a sampling plate, which is the PCR plate that you see on the right. And in that change of format is fundamental to being able to do large numbers of samples and keeping track of those samples. So I'll show you how that's done in a second. The other strategy that we adopted very early on, right at the beginning when it was being talked about, was a strategy of pooling. And so pooling is a process that allows us to combine multiple samples into a single test, and then test that pool, and then go back and identify the individuals that made up that pool and get back to an individual test. And pooling is a very popular strategy now. It's been around for a long time. In blood banks have been using pooled samples to make things possible and to save time and money for a long time. And so that's a strategy that we decided to adopt very early on. Next slide, please. So there were many challenges that we faced in setting up this lab. One of them is that, so if you look on the left-hand side, those are the tubes once they get out of the bags. And so one of the things that we have to do is we have to be able to position every single sample as it's going to go into those assay plates that we saw on the previous slide. And this is accomplished by sort of an ingenious process where we have those silver racks there. Those aren't silver, they're actually aluminum. And those racks, every one of those holes holds a tube that gets, because it has a barcode on it, it gets scanned into a particular position, and that position then it translates into one of the positions in those plates that we saw earlier. And so that's called racking. And one of the challenges that we faced right from the beginning was that none of the instruments were actually set up or had racks that would fit the tubes that we could get. And so thanks to the manufacturing people over at the Olson Center, we could custom make racks for any different kinds of tubes. And that's the UNH logo on their manufactured racking tubes. Over on the right is how those samples are pipetted out. It's actually, interestingly, the pipette that Timothy was using is the exact same pipette. Ours is just attached to a robot instead of to a TIM. And it works really wonderfully. And so we're able to, in a matter of 5 minutes, go from 96 samples placed out in these racks into the 96 well plate format that then is compatible with all of the downstream robotics. This seems like seems kind of trivial, but this is what allows us to be able to do the thousands and thousands of samples per day. Next slide, please. So I mentioned that we have a pooling strategy. And so basically what we do is we make up 4 of those plates and we combine them all together into one plate, which saves us a lot of materials, especially at a time when the supply chains were limited. And of course, it reduces the cost. And even more importantly than either of those, it actually reduces the amount of time. The downstream steps of the assays are also time consuming, and there are multiple. You have to process the samples to extract that viral RNA from the coronavirus, and then you have to assay the samples to identify if there's coronavirus in the samples. Pooling, if anybody has actually read about it, has some potential drawbacks. The most important one is it only works when the infection rate is reasonably low, and so when the vast majority of your samples are actually negative, and so it does have that limitation. And thankfully, when our students and staff came back on campus, the infection rate was very low and it's remained low, which is wonderful. Next slide, please. So as Marian mentioned, this is kind of an interesting thing. I've been at UNH, I was hired here as the Hubbard Chair to bring technologies like this to campus. And mostly that has been in support of research. And we've had some large scale success, but I have never had the entire congressional delegation come to campus, let alone the governor and dignitaries like Ambassador Birx here. So this has been a little bit shocking and wonderful to have that, and in particular for the staff at the lab. They really see the effects of their very hard work. Next slide, please. So just want to acknowledge, what has been the keys to success thus far. We're not finished by any means, but the keys to success thus far. And one, I give a lot of credit to Marian and her leadership in setting up this task force and bringing a group of people together and working very, very hard to make this happen. The other group that I have had a chance to work with and have made so many contributions and really keep the wheels going on this operation are the staff that were hired. And Rick Cody and Tamara Wood did all of this hiring, and I can't tell you what a pleasure it is to work with this group of staff, and their dedication to this has been incredible. The other part of this that I didn't talk about much, but you can probably imagine is really very important, is keeping track of all the samples. And we couldn't have made any progress in that without Tony Westbrook, who's a computational scientist here in the Genome Center. And so with that, I will close and take any questions for us. Thanks.
Thanks, Kelley, and thanks, Tim, for those excellent presentations. So now we'll start the Q&A portion, and let me just remind you that if you have a question, you can enter it into the Q&A tab at the bottom of your screen. Just click on where it says Q&A, and we'll try to get your questions answered. Okay, so I think this, either Tim or Kelley can answer this. What's the difference between a BSL-2 and a BSL-3 hood, and why did you need one or the other? Want to take that, Tim?
Yeah, sure. Depending on, I guess, the level of danger in the virus or the pathogen that you're handling, that will dictate the environment that you work within. And every institution or company or whatever has a committee that helps decide where these things should be handled. In part, like our government and the CDC and National Institute for Health can kind of help guide this as well. But typically, like the higher the number, the more dangerous the pathogen. So like Ebola virus is handled in a biosafety level 4 laboratory. And so that environment is it's for things that are really dangerous. They have like a high kill rate, and there are no known therapeutics or vaccines or anything. So we work one step below that at biosafety level 3 with most of the coronaviruses. But like, you know, and Kelley can comment on the environment that they handle their clinical samples, which I would imagine is biosafety 2+ or something like that, which but basically, the number and the biosafety level is directly related to how sick you can get from the thing you're working with.
Yeah, that was a major challenge in setting up the lab, was, you know, having the BSL-2+ hoods installed and inspected. And basically, anytime the virus is exposed to the air, that before it is inactivated, that has to happen. There are some interesting stories, because when we set up the lab, we couldn't, the biosafety hoods wouldn't fit through the door. And we're on the 2nd floor. So there are pictures circulating out there with a two giant biosafety cabinets going through a window to get into the building.
Oh,
so
awesome.
We spared no effort to
like
set
call
that up.
Incredible Hulk to just like break the wall open.
It felt like it.
We were lucky to have a space that was already set up as a lab that had been vacated in health and wellness. So that was a real advantage to us. But we hadn't figured out having to hoist the BSL-2 hoods in by a crane. So so the next one is for Kelley. Kelley, what's the turnaround time for the testing? How long till you get an actual result from the time, say, that the student drops off the sample?
Yeah, that's a good question. So the way the students drop off the samples in drop boxes around campus, they are picked up at 9, 11, 1, 3, 5, and close to 6:30 p.m. The samples that are, for example, that were picked up at 9 this morning, they are probably well on their way to being assayed. And at this point in time, almost everything is processed within 24 hours. And so those are the vast majority of cases fall into that. There are cases where, because of the way the pooling goes, you may be delayed overnight to get your results. But and you know, if your tube was labeled correctly and got in there, that's a pretty fast turnaround time. We also retest things that are brought to us as special cases, and those are processed in the evenings and are completed in about 5 hours.
Do you have 3 shifts in the lab?
No, we only have 2 shifts.
But that's pretty fast.
Yeah, but also 6 days a week.
Wow.
Yeah.
So, so this question is for Tim. Tim, do you think it's more likely that coronavirus will ultimately be managed long-term via antiviral medications or vaccines?
That's a great question. I'd say we probably need both. You know, I guess it's on the vaccine front, you know, vaccines are only good as long as a certain number of people are using them. So if we don't get the coverage we need to kind of tamp down the virus in the population, then we may be leaning more on antiviral drugs or antibodies or like things like that. So I think we need everything that we can get to win against this thing. So.
Okay, thank you. Let's see, there's suddenly a flurry of questions. So, so Kelley, can you explain again, in just a little bit more detail, the process of pooling? Is the integrity of each individual sample preserved through the entire process? How does that work?
Yeah, I should have given more explanation there. So, the individual samples are maintained throughout the process. So we have when we pipette the sample from those tubes into the 96 well plates, if you will, that those are as individuals, right? And so there are 4 plates that are produced of individual samples, and then they are combined in the assay and assayed as a pool. And at the end of that, what you typically have, or the vast majority of assays, are negative, and so nothing happens to those samples anymore if they are negative. In the few cases where the pool of samples tests positive, then we can go back to those original specimens, which are intact as individuals, and assay them individually. And in our case, because we're pooling at a level of 4, it's always one of those 4 individuals that made that pool positive. And then we actually go through a whole nother round. We ask the person to come in and be tested again and immediately. And then we run those every evening.
So someone wants to know if you're concerned about pooling the samples, diluting them so much that it decreases the sensitivity of the test.
Yes, I mean, that is always a question. And you are diluting. That's part of what led us to use this very conservative pooling strategy of 4. Other places, you know, have demonstrated the value of this pooling, you know, at higher levels of pooling. We also, at the beginning when we were working on this, did a lot of validations with the state, providing us samples that were detected at various levels of virus concentration. And so we spent a lot of time making sure that that we were minimizing the effects of this dilution on our samples.
I mean, you could probably, you know, dilute, say someone's positive, you could dilute it 100,000-fold and it would still be caught in Kelley's test. So diluting something by 4 is like, for me anyway, unless the sample's like really low to begin with. So
Yeah, I mean, the validations worked out really well, but there's always those cases where, you know, well, the ones that we're concerned about are ones that are really early in infection,
Right.
where the virus titer might be lower. You know, the other thing, and I have to give Marian credit for this, is the frequency of testing. If we miss you because it's early, we're going to get you in 3 days. So we test a lot, and that's been very valuable.
We do test a lot. So thanks, Kelley. So I'll go back to Tim. Tim, why are zoonotic diseases on the rise? Or if they're not really on the rise, why are you so worried about more of them popping up in humans? And also, according to your cartoons, it looked like the virus went from bats to humans. Can you comment on that?
Yeah, great questions. Bats, basically anywhere you can find bats, you can probably find bats with coronavirus in them. You can find coronavirus in bats in North America, Mexico, Southeast Asia, Europe, you name it. Anywhere, any continent where you have bats, I pretty much guarantee you can find bats with coronavirus. They just seem to be really good at harboring these viruses, apparently to no ill effects. Like, they don't seem to be sick or anything like that. So they seem to be a really good reservoir for coronavirus, and it's believed that the original SARS coronavirus came from a bat, as well as MERS coronavirus in 2012. It's now endemic in camels, but it's believed to have originally come from a bat as well. So, but regarding like it seems like zoonotic diseases are on the rise, and really, as we change how we grow food and expand as humans into places that normally aren't places, they're normally not places where people live, we are exposed to things that we haven't ever been exposed to. And I think, what drove the emergence of SARS-CoV-2 is probably like an amalgamation of lots of human, virological, ecological, environmental factors that are hard to really point the finger at one thing. But I think, that makes me, it kind of gives me pause as, we expand as human beings and as the human population expands. Like, what can we do to try and minimize this, since it seems to be something that we're maximizing right now? So I don't know.
You said in your GQ article that that you had hope for that this experience would change things, how we did things on a grand scale. What types of things did you mean you hoped would change?
Yeah, and that was more like based on like how people do the things that I do. Basically, like looking at all the cool tools that Kelley has and how you got your testing up and running pretty fast. Like, those are the things that, once you all learn how to do that, if you ever need to do that in the future, there's already a roadmap for this, right? So that's kind of what I was getting at, and also my children have a good working knowledge of virology and epidemiology, and they're like in first grade and third grade. So I think globally, we've basically been forced to take a virology and public health class. And I think moving forward, we as humans will be better prepared for another emerging disease, just because we're going through this thing together right now. It's insane.
Yeah, that's a good segue into my next question for Kelley, which is, have you collaborated with other university labs? What other labs at universities are doing things similar to what we're doing? And what do you see for the future uses of the lab? So who else? How are you working?
Yeah. So I wouldn't say, I wouldn't call it as much a collaboration as sharing some ideas. Everybody has been running as fast as they possibly can. And you know very well that we we here at UNH have as well. But it has been very useful to talk to some of our other universities. It started really with a group interestingly enough, at the University of Vermont that I have worked with for a number of years. And that was critical in understanding which of the tests, both emerging and established, that we chose to go forward with. We have since shared our experiences very freely with other institutions in both Pennsylvania and in Boston, the Boston area. And we've in my recent workshop, we had, you know, the University of Massachusetts at Amherst, and who, you know, everybody is doing things differently. We sort of take the cake when it comes to the number of tests that we're doing and the frequency, but everybody is strategizing in ways with a common goal of keeping their campus communities open. And so it's been very important. I would say that the most important collaboration that we've had is, in fact, the collaboration with our public health lab. They've been extremely supportive. It actually started, interestingly enough, very early on, them asking us for help. And then we turned the tables on them, and they have been very helpful. The system that we set up is in parallel with the system that they've set up. And so that's been a very good both intellectual and sort of physical collaboration with them.
And what do you see for the future of testing in the lab?
Well, I'm right in there with Tim on this. First of all, I don't see this going away over Christmas. So we have an immediate future, and probably a bigger responsibility than we're taking now. But I do think there's a long-term need to have the capacity to do this. And, you know, we have a very, very strong veterinary diagnostic lab whose expertise is in the emerging zoonotic diseases. We have a very strong microbiology faculty whose expertise is in line or directly the discovery of emerging pathogens in the environment. So we have a synergy between faculty research expertise and also the Genome Center is I believe a key to that ability to move forward and to innovate. It may not always be the way we're doing it exactly, but everything from the informatics that's been built around this to the instrumentation and the training of staff and students in this area, I think it builds a fundamental capacity here that I wish I could say we won't need it, but I suspect, you know, we will.
Thanks, Kelley. So Tim, you talked a little bit about drug testing and drugs moving from phase 1 to phase 2. Can you talk about the meanings of the different phases? And are you concerned about a vaccine being rushed through the system so quickly?
Yeah, okay, so the first one, what do the phases mean? So like when you go into human clinical trial, the first phase or phase 1 is always in healthy people who aren't sick to see if your drug is safe and is tolerated in a healthy adult. And once you establish that the thing that you're trying to use as a medicine is safe, then you move into a sick population. So that would be, for example, COVID patients. So first you test in non-COVID patients, and then you test in COVID patients in phase 2 to establish whether or not your thing is working. So you can do a placebo-controlled trial where some people get your medicine and some people don't, and that way you can tell whether or not your thing is having any kind of benefit. And then phase 3 is basically an expanded form of phase 2 typically, where you're looking at drug efficacy in bigger populations or diverse populations like different ethnicities and stuff like that. Am I concerned about the rate at which the vaccine process is moving? Not really. I think one thing that money can get you is you can do multiple things. Like normally it'd be like an iterative process where you do one thing and you would take a pause and see if it was okay while you're, and if you do that, then you ramp up production of your vaccine. Everything's being done at the same time. And that's why I think we're winning as far as speed goes. But I don't believe that the companies and the government would take and cut corners safety-wise just to get a product out there. And I would take the Moderna vaccine any day of the week.
Good to know and reassuring. So, so Kelley, how expandable is what UNH is doing to the broader community? Is there a reason to consider trying to apply the type of testing that we're doing, say, to the K-12 faculty and students or the broader community?
Yeah, I mean, I think there is. I hope that that's in our future. We set up a lab in a very short period of time, and you know from our discussions, and you know that there have been many days when I'm not sure that we're going to make it, and as time has gone, I think we do understand how we can move forward to make a more resilient and robust version of what we have. And I think as we move toward that, then that's gonna bode well. The field is changing as well. That the exact tests that we're doing at some point that might become not the optimal, but most of the things that I was talking about as keys to the success aren't really which PCR reaction you're doing. It's do you have the capacity to go from those tubes, whether they're nasal swabs or saliva tubes, into an assay format, and that's where the lab actually has been really good and made a big difference. And pooling isn't just an RT-PCR process. It's going to save money and time, even more importantly, time in all different kinds of assays.
Thank you. Tim, can you explain the difference between the virus being airborne versus aerosol? And if it is spread by droplets or aerosols, shouldn't I just be able to cover my mouth or nose when I sneeze and prevent the transmission of that?
Yeah, that is a really hard question. So I think when people say airborne and aerosol, they're kind of like exchangeable terms. I guess like depending on when you sneeze and cough, lots of different sized particles come out of your mouth. And if the virus happens to be on the smallest particle, which is like an aerosol, then it tends to hang in the air more and travel further away. It's also harder to contain. So like if you were to take a close-up picture of even a cloth mask, it's essentially a screen, right? And, it can stop the big droplets and help slow the small ones, but it's like not perfect, but still, I think it's hard to contain these small particles that you can't see if you're coughing into your arm or even a handkerchief. So that's why, like, whenever I go out in public, my door's closed now. Whenever I go out in public, anywhere, I wear this because I think this is it looks cool. And I think this is our best chance to help slow virus spread amongst our family members, colleagues, and stuff like that.
Thanks, Tim. Well, I'm looking at the clock and there are just so many questions, but we're nearing the end of our time. So I wanted to just take a moment to thank, Kelley and Tim for excellent presentations, and thank the Alumni Association for having this wonderful event. You can look for a follow-up from the webinar or after the webinar from the Alumni Relations team with more information about this event and other upcoming programs. And we hope to see you again soon at the next one of these events. So we really appreciate your attendance and your support, and hope this has been educational and interesting for you. So thank you all.
Thank you, Marian. And thanks to our students. I forgot to say that. They're really doing a great job.
They are indeed.
Go Wildcats.